Hello and welcome again. So now today we were discussing about this innate response, immune responses towards the viruses, and among that we have actually discussed about the innate immune responses to the bacteria previously. Now we are talking about the innate response to the viruses, in which we have to talk about the role of the interferon in the previous lecture. We will be talking about this NK cells today, and these NK cells are actually the member of the innate branch of the immune response. They exhibit the capacity to kill the viral infected cell and tumor cell. So you have to understand this NK cell can kill this viral infected cell and tumor cell. How they can kill? They kill via the same mechanism of inducing apoptosis observed with this cytotoxic T cell, that is granzymes and the perforin. So they kills by the same mechanism by which this your cytotoxic T cell kill, that is inducing apoptosis in observed with this say cytotoxic T cell by the granzyme and perforin. NK activity is increased in the presence of interferon, that is alpha and beta and interleukin 12. So NK cell activity is increased by interferon that is alpha, interferon beta and interleukin 12. So this three thing is there to increase the activity of the NK cell, and they induce the apoptosis by this cytotoxic T cell, that is the granzyme and the perforin. Now talking about how they kill the cell. NK cells share a common early progenitor with the T cell, but they do not develop in the thymus. They do not express antigen specific receptor outside CD3. The marker usually used to enumerate this NK cell is CD16 and CD56. So that you have to understand. We are talking about the NK cell that are part of the innate immunity system. They kill this viral infected cell and tuber cell by the same process by which the cytotoxic T cell kill, that is granzyme and perforin, and they are activated by this interleukin alpha, interferon alpha, beta and interleukin 12. They are the part, they actually originate from the lymphoid cell, but they are not part of your adaptive immune system. They do not produce any T cell CD3 marker. They actually process this CD16 and CD56, which is the marker that is helps to recognize this NK cell. So if there is anywhere mentioning about the CD16 or CD56, you have to understand we are talking about this NK cell. Their recognition on the target cell, target is not MSC restricted, and their activity does not generate any immunological memory. So there is no immunological memory. Obviously they are the part of the innate immune system, so there will be no immunological memory, and they do not have MSC restricted, like MSC1 act with the cytotoxic T cell and kill the viral infected cell or tumor cell, and talking about the CD4 that will act with the MSC class 2 and then help in the antibody production. So this is not related with the MSC, but they are related to the one important thing that is called this killer activating receptor and killer inhibitory receptor. So NK cell employs two categories of receptor. One is killer activating receptor and another is killer inhibitory receptor. This, if only killer activating receptor are engaged, the targets will be killed. So if only one is found, that is killer activating receptor is binding, then it will kill the cell. But if both killer inhibitory cell and killer activating cell are ligated, then target cell lives. Therefore the inhibitory signal trumps the activating signal. So the inhibitory signal is actually powerful than the activating signal. So if inhibitory signal, there is a binding with the inhibitory signal. Let me show you over this picture. You can see over there, this is the this is inhibitory receptor, this is activating receptor KAR, and killer inhibitory receptor and killer activating receptor. If there is killer inhibitory receptor is binding with the in targeted cell, there is the proper functioning, then this cell will not be killed. If this killer inhibitory cell will not be binding, only killer activating is binding, then this cell will be killed by NK cell. So they actually see the similar structure. If the familiar cell is found, so this will not be killed. If there is something difference in the familiar faces, there is abnormal faces, then this will be killed. Like if you can remember as a dog, when the dog in your house say your similar faces, they are happy, they are not biting anyone. But if they are finding any asimilar faces, new faces, then they will kill that organism. They will bite that say new people. Similarly, this NK cell will kill that cell if they are found new protein or say new stressor molecule on the cell surfaces. This is the way how this NK cell is acting. So these are the two receptor, killer activating receptor and killer inhibitory receptor. We have talked about this, and if only killer activating receptor is bind, the cell is killed. If both receptor is bind, cell is not killed. So NKG2D is the major killer activating expressed by NK cell. There are many ligands for KR. The MIC glycoprotein are one type. MIC protein are the stressor protein that are expressed only when the cells are infected or undergoing transformation. Upon binding to the killer activating receptor to this stressor or say undergoing dying receptor protein, NK cell becomes cytotoxic, resulting in the death of the cell. So this killer activating receptor bind to the infected cell protein or a stressor protein. If the cell is infected or undergoing transformation, they will release this MIC protein. This MIC protein will bind to this killer activating receptor of NK cell, and your cell will be death if only binding to this killer activating receptor. If they are binding to the killer inhibitory receptor, they will not be killed. Why? Because killer inhibitory receptor activate protein kinase phosphatase, we inhibit the intracellular signaling activation by removing the tyrosine residues from very signal molecule. The killer inhibitory receptor on the NK cell bind to the specialized type of MSC class 1 antigen called HLA-E. HLA-E has a unique tissue distribution as do the other class 1 HLA molecule. This HLA-E molecule bind to the peptide derivative from the leader sequences of HLA-A, B and C. HLA-E requires a bound peptide for the proper expression within the cell. Therefore, the amount of HLA-E expression indicate a cell over well-being. That is important. This HLA-E, if that is present on the cell, that's there. This indicates the HLA-E expression indicates over well-being of the cell. So if there is a HLA-E present on the cell, means the cell is totally fine. The cell is totally active. The cell has no any problem. So this HLA-E, if present on the cell, means that your cell is fine. This is a similar phases. This is okay. That is detected by this killer inhibiting receptor. So if HLA-E is okay, if HLA-E is there on the cell, then they will bind to the killer inhibitory receptor and the cell will be not be killed. That you have to understand. And when this HLA-E will be present on the cell, if the cell is fine, if the cell is well-being, if the cell has no problem. So if the cell is okay, why we need to kill our own cell? And what indicates the cell is okay? The presence of HLA-E on the cell surfaces. And if there is HLA-E on the cell surfaces, that will be bind with their NK cell inhibitory receptor, and your cell will be not be killed. So that you have to understand. These are a viral infection or transport cell. The amount of class 1 HLA expression may be decreased, which will prevent the leader sequencer from binding to HLA-E. This will decrease the expression of HLA-E and make the cell susceptible to NK mediated killing. So if there is cell is infected with the virus, there will be a decrease in the HLA-E on the cell expression on the cell surface. So if HLA-E is decreased, means cell's well-being is not there. If HLA-E is present, cell well-being is there. So since there is decrease, cell well-being is not there, now NK cell can be killed. NK cell can kill that cell. They have their freedom to kill. If HLA-E is present, you cannot kill your own cell. But if HLA-B is down-regulated, decreased, means now cell is diseased, dying, infected. Now the NK cell has authority to kill that because it is not binding with the inhibitory receptor. Now this killer activating receptor can only bind, and that will kill. So this you have to see. This is where we are talking about. Okay. Now there is another mechanism by which NK cell can kill, and that is interestingly when NK cell are activated through the FC, that CD16, only one signal is required because the antibody signals that there is active infection. So this occurs through a mechanism called antibody dependent cell mediated cellular cytotoxicity, ADCC, that is antibody dependent cell mediated cytotoxic. This is actually dependent on the antibody. So this is antibody mediated, and that will bind to the CD16. So NK cell has two receptors, CD56 and CD16. If they bind with the CD16, they will directly kill that cell by ADCC. If they bind with the CD56, which we have talked, killer activating receptor, a killer inhibitory receptor, and kill the cell. So there are different mechanisms. Let's revise again this killing. There are the killer inhibitory receptor and killer activating receptor. If killer inhibitory receptor found this well-being of your cell indicator that HLA-E is present in sufficient amount, this cell will be not be killed. If this HLA-E is decreased, then obviously this stress molecule, MIC molecule or stress molecule will present, and that will be, this cell will be, there is no HLA-E is not there in the infected cell, that is in decrease, so that will not bind to the inhibitory receptor, but the stressor molecule will be there, that will bind with your killer activating receptor, and NK cell will be killed with this granzyme and porphyrin, and your all viral infected cell and tumor cell can be killed by this way, by your innate immune response or say NK cell. So let me give you one more important concept. The cells are actually viral infected cell are killed by this NK cell. If there is if they cannot be killed like this, then there will be the cytotoxic T cell which will kill. By the formation of that will be the adaptive immune response. Okay, and if that will also not be there, then there is the ADCC mechanism. So there are three main mechanisms. NK cell can kill by this killer activating and killer inhibiting combination. Then MSC1 class will be actually bind with the CD8 cell, and this by this way your viral infected cell can be killed, and also this ADCC mechanism, NK cell can kill by your ADCC mechanism. So viral infected cell can be killed by the three important way that you have to understand. So hope you have understood the role of your NK cell and also how the viral infected cell are killed. Thank you.